All right. So today will be day 4. We're still talking about Unit 3, and this will be our test review, our Unit 3 test review on cells. Okay, so this will be the last video for the cells unit. So day 1, remember day 1 was mainly talking about you need to be able to make sure that you can compare and contrast the differences in different types of cells. That was the main focus on that day. So that would be prokaryote, eukaryote, as well as comparing the different eukaryotes such as plant, animal, and fungi. But I also might ask you to compare some of those specific cells, like an animal cell to a bacterial cell, or a fungal cell to a bacterial cell. So you're going to need to make sure that you know enough about those individual ones to where you can answer those type of questions. So, of course, first off, knowing the difference between a prokaryote and a eukaryote. Um, Prokaryotes do not have a nucleus. What are the different types of eukaryotes? The different types of eukaryotes are going to be your plant, your animal, your fungi, and your protist. We're going to mainly focus on the plant, animal, and fungi. That's where most of those contrasting questions come in at. Which eukaryotes do not have a cell wall? That would be the animal, and that would be the protist. Which do? Now we're specifically talking about eukaryotes. So that's going to be the plant and the fungal. Now the differences between those cell walls is the plant's made of cellulose. The fungal cell walls are made of chitin. Which eukaryotes undergo photosynthesis? We're going to mainly be focusing on the plant cells. There are some photosynthetic protists out there, but that's not where our focus is going to be. We're going to go with that, what's that general characteristic that we typically see a lot of. What does it mean if an organism is a heterotroph? That means that it doesn't make its own food. It can't get its energy from internal mechanisms. It actually has to go out and consume, or it could be like I said, like a consumer, like an animal, or it can be a decomposer, like fungi or decomposers. Fungus is a decomposer. How does a fungus obtain its nutrients? Like I just said, it's a heterotrophic decomposer. What are the 4 structures that all cells have in common? So, like I said, whenever you're trying to figure out similarities and differences between on questions, by knowing number seven's answer here, that could really help you out in discerning what's the correct answers. So, the 4 similarities that all cells have are going to be cell membrane, ribosomes, cytoplasm, and DNA, not nucleus. A lot of students get confused whenever I say DNA. That means I'm saying nucleus. That's not correct, because remember, bacteria do not have a nuclei or do not have a nucleus. What are the differences between a plant and an animal cell? So we're talking about the big differences here. Plant cells, they have a cell wall, a large central vacuole, and a chloroplast. Animal cells, on the other hand, have a lysosome, um, and centrioles. What's the other name used for prokaryotes? Bacteria. So I can sit there and ask questions using the term prokaryote or bacteria. Those two are synonymous since they are the same types. And then we've actually already contrasted prokaryotes and eukaryotes, but I'll go over it again just real quick, and I'll focus on the prokaryote side. Prokaryotes do not have a nucleus. Eukaryotes do. Prokaryotes, because they don't have membrane-bound organelles, they're very small. The only organelle it actually has on the inside is going to be ribosomes. All right, so that takes care of a couple of them right there. Um, Prokaryotes are actually a lot older than eukaryotes as well. Their DNA, they only have a singular strand DNA, and it's circular as well. Whereas eukaryotes, we have all of ours bundled up into chromosomes. Day 2, this is basically talking about the functions of the different organelles. See if you can sit there and label out these functions. Just take a second, pause it, see if you can if you can answer these real quick. All right, so I'm about to give you the answers. Number one, cell membrane. Number two is the plant cell wall. Number three is the Golgi. Number four is lysosomes. Number five is chloroplast. Number six is cellular respiration. Number seven is mitochondria. Number eight is the cell membrane. You could also say plasma membrane if you would want. A on the right side, so we're on A, is the endoplasmic reticulum. B would be the nucleolus. C is the nucleus. D is ribosomes. E is rough ER. F is smooth ER. G is they're located in the nucleus. H, cellular respiration is the process of making ATP, and that happens at the mitochondria. I would be the cytoplasm. That's just kind of like a real quick check to see if you know some of those basic facts. So if I were to tell you that if I had a question on there, and let's say I'm looking at letter H, where it talks about cellular respiration. I'm telling you that, man, this something's happening inside of a cell is starting to malfunction. It's no longer able to undergo the chemical reaction of cellular respiration. Which organelle is malfunctioning? So that's just a backwards way of saying, okay, do you know that cellular respiration is done by the mitochondria? If cellular respiration is no longer happening, then that means the mitochondria is malfunctioning. And so that's a general type of question that you might see. That's kind of a backwards way of asking a question. Instead of just saying, what does it do? That's just another way of making it a little bit more wordy. Day three, still talking about some of those structures and their functions. Add support throughout the cell. On the inside, we're talking about the cytoskeleton. Only in animal cells for cell division. That's going to be the centrioles. Number three is flagella. Number four is cilia. Number five is pseudopodia or pseudopods. The three methods of cellular motility or movement or locomotion, that would be the flagella, the cilia, and the pseudopods. And letter B would be the spindle fibers. Again, so I just put this one on here to where, so I've been asking a lot of questions about them and then having you fill in this part of it. Make sure you can work backwards with it. I might just give you the term and make sure that you're able to think of it that way. Hopefully you've already been doing this on our labeling exercises as well, but this is just another way to hopefully help you to do that. You can sit there, hear the names, pause it if you want to sit there and go through there and see if you can list them out, and then go back and look at your notes to see if you got it right, see which ones you're getting wrong. Don't forget, I also have quizzes available to you on this to where you're going to see a bunch of different style of questions. That'll be a very good thing to look at. Again, these same images, the same diagrams, you could pause it here. I'll have the answers on the next one just to give you time just to rewrite it and make sure that you know there's no reason on this test not to do very well on it. Okay, moving on. There are the answers. Moving on. There you go. You can pause this, give yourself some time to write it down. Make sure you know the functions that go along with it. So I was saying on the other day that I was going to go over some style questions that you might see. So J over here. Now let's say this is a cell. This is this is a question. This is the diagram that I use, and I say in the question that basically, what's the function of J? Well, that means not only do you have to know what J is, but then you also have to know the function of it. Because I might not have listed out chloroplast and what it does. I might just have it undergoes photosynthesis, or it's capturing sunlight to make glucose for a plant cell. So you'd have to read through those questions and be able to discern what it is. I could also ask you which organelle is malfunctioning if a cell is no longer able to package and modify materials for transport. Well, that becomes a loaded question. You're going to need to make sure that you know that if we're talking about packaging and modifying, that we're talking about the Golgi. And then you have to be able to identify the Golgi. In case you're wondering, the Golgi is letter I down here at the bottom left hand side. It's letter I. So that would have been the correct answer there, would have been letter I. So that's kind of like a two-part question. So make sure you can do those style. Again, there's the answers. You can pause here in case you need to check yours. And then lastly, here's this. Same type of thing as on the other one. I could sit there and give you this image and say and ask you about the differences, kind of going back to day one stuff, and I could just say, hey, which two organelles, or which organelles does this cell have that a plant cell doesn't have? Well, first you're gonna have to make sure you know this is an animal cell, and 3 and 6 are those two, because 3 is the lysosomes and 6 is the centrioles. So that's a way of taking the same type of question, but adding a diagram to it, and it adds another layer of complexity. So make sure you're ready for those type of questions. And like I said earlier, and here's your answers for that one, for the animal cell. Like I said earlier, you do have those quizzes available to you, so please use those. Just sit there and just give yourself some time to go through it. All right, and there you go. That's the end.